Hello and welcome to MMI. In this series of tutorial videos, we'd like to show you the basics of the MMI CellCut and how to get good results for further research. The MMI CellCut is our laser microdissection system designed to cut tissues, large regions of interest or single cells, and even living cells for further processing. With its high-pulse UV laser for precise cutting and the patented CapLift technology for reliable tissue and cell isolation, it will help you with all your laser microdissection needs. In this video, we show you how to ablate your sample and talk about the differences, pros and cons compared to normal laser cutting. If you want to ablate structures, first you have to check the checkbox for ablate and then ablate with contour. Note that the ablation mode has its own set of laser settings. Maybe you want to use some different settings like defocusing the laser a bit and turning up the laser power. Then you can draw the shapes as usual. In this case, we want to ablate a circle. Then click on Cut and the system starts working. But this time the system will not cut out your shape, but ablazing it. The distance between every row is defined by the spacing value. And if you have checked ablate with contour, in the end the system will also cut around the edges of your structure. Notice that the cap was down, but instead of a collection cap, we used a normal reaction tube and put some water into the lid to collect the debris from the burned material. Of course, the cutting will also work with the cap up. The main difference between laser cutting and ablation are obvious. One will just cut out one structure, the other will completely destroy it. But while laser cutting and collecting needs a membrane slide, the ablation mode can also be used on all the samples that are mounted on a glass slide. If you're done ablating, don't forget to uncheck the checkbox. Your colleagues will thank you. We hope this video helped you with working with the MMI CellCut. Feel free to visit our other videos for more information about MMI instruments and handling.